Hi everyone, Brenda Gustin here, and we're going to start eukaryotic cells this week, and I'm in section 4.2, and I just have a little bit of information to give you in this section. I wanted to remind you that there are prerequisites for this course, and one of them is a biology class, and somewhere in that biology class you should have learned about cells. Now, typically what we learn about when we learn about cells is we learn about the eukaryotic cell. And then we kind of learn about prokaryotic cells later on and some of the variations. So we've already talked about prokaryotic cells. And I just want to remind you, if you're rusty on your eukaryotic cells, that you might want to go back and look at them. There's a wonderful diagram right here, and you can even click on different things and learn about them. But here's reminding you what some of these cell parts look like in eukaryotic cells. I'm not going to quiz you directly on eukaryotic cell parts, except for the ones that we mention as part of lecture. I'm going to have some assumption that you know what a mitochondria is and what it does, what a nucleus is and what it does. So you can go through that. The book goes through lots of really great information on all these different cell things. We're going to mention cell walls. We already talked a little bit about cell walls this semester. But when we talk about fungi, we're going to talk about how they have chitin in their walls. And you might remember that plants have cellulose in their walls. Animal cells don't have cell walls. So all that should be a review. One of the things I wanted to mention, though, that's pretty important is down here, we talk a little bit about ribosomes. And hopefully right now you can say, oh yeah, I know what a ribosome is. Actually, we can go look at, if I back up, I can look at some pictures of ribosomes. There's some on some little dots there. But there's some dots there that are ribosomes on the ER. And remember that there's lots of ribosomes floating around the cytoplasm and on the ER. Now remember that prokaryotic cells don't have endoplasmic reticulum, but they do have a lot of ribosomes floating around. Hopefully you remember very quickly that ribosomes are the site of protein synthesis. Remember, proteins are one of our four major groups of organic molecules, and they're absolutely essential for the normal functioning of any kind of cell, whether you're eukaryotic or prokaryotic. So ribosomes have to function. You have to be able to make proteins to survive. One of the things they talk about in this paragraph is that prokaryotic ribosomes are actually a little bit smaller. They have a little bit, even though they do the same function, they're where RNA is used to make proteins, they have a little bit different structure. And because of that, there actually are, here it says, this difference means that we can use antibiotics that target prokaryotic ribosomes without harming our own eukaryotic ribosomes. So that difference gives another target for drugs. Streptomycin and tetracycline are two antibiotics that can stick to bacterial ribosomes and stop protein synthesis. So again, when you're developing drugs, you're always trying to look for what's different in the infectious agent compared to the host, and what can I damage in the infectious agent without harming the host. And so ribosomes are another good target. Do you remember the other target that we had in a previous lecture? Should I give you a hint? Has something to do with peptidoglycan. Okay. The other thing is just down in this table here, it gives you another little summary, and it does say with ribosomes, let's see, where do we go? Well, there you go. Ribosomes right there. You click that little thing, and it says bacterial, archaeal, prokaryotic ribosome is functionally similar to the eukaryotic, but structurally unique. So, like I said, they do the same things, but they kind of look different. So they can be a target for drugs. And that's where I'm going to stop right now. Good luck.